PROHITS, prokaryote proteomics at high temperature for single cells. Can you briefly introduce the PROHITS MSCA project that you are coordinating and its main objectives?
I certainly can. So the MSCA project, essentially, this is a doctoral network. So its main purpose is to train young researchers around a particular topic that is often interdisciplinary, that connects different disciplines together. So the PROHITS MSCA doctoral network is aimed to basically develop techniques to study small cells, prokaryotes, bacteria and archaea that live at very high temperatures. So some organisms live at very high temperature, and we don't really understand these organisms very well. So what we're doing within the PROHITS is developing techniques, a technique called proteomics, where you can study all the proteins and molecular machines that make cells work within those extremophiles, as they're called, so those cells that live at very high temperatures, and to then try and understand how they can operate at these high temperatures. The further goal then is that once we understand this, then we can adapt these organisms and then you can use them in the bioindustry. So essentially what you do there is that you adapt these organisms so that they produce certain molecules, for example, drugs or something to do with biofuels, where it's an advantage if they can operate at high temperatures, because if you grow them, they generate heat. So if you can have organisms that operate at very high temperatures, you can do much quicker reactions, essentially, and you can also then make the whole bioreactors much more efficient. So that's the aim of the project. So we try and understand proteins that operate at high temperature. We understand how these proteins all work together within the proteome, so all the proteins that work together within the cell, and then we try and adapt the cell to be used in bioindustry.
What inspired the conceptualization of this project?
The project started from talks between myself and Lennart Martens, who's one of the PIs on the project, and they work on proteomics. So they look at cells and then they look at which proteins are inside the cells, and also how these proteins can be modified. And we've already been thinking a lot about different ways on how we can collaborate together. And one of them that came up also because of a collaboration that I have within the VUB with Evelien Peters, who's also a PI on this project, who works on these extremophiles. So she studies them more from a biological perspective. What we've tried to do is combine these two elements together, and what we've done there is to then bring together people that also do experimental proteomics, so that do the studies of these cells, also try and study them single cells, so separate the cells from each other so that you can look at them one by one, essentially study how they vary, how these cells are different from each other, as well as then incorporating genome scale networks, which are essentially models of the cell and how it works. So all these things integrated were essentially what came out of this conceptualization. These were the things we needed to bring the concept of trying to understand these cells and using them for bioindustry to fruition.
What were the key steps involved in developing the initial concept of the project?
When you start a project like this, it's always really important to get the right team together. This is essential. If you have the right people, then the rest becomes much easier. So besides Evelien, Lennart and me, we all have our own connections. So we try to identify within the concept that I just described, we try to identify the right people that we already knew, ideally, because then you know that they're good to collaborate with for the project, and to then draw them in. So explain the concept and then try to get everyone enthusiastic about the concept, which was not too difficult in this case, luckily. And then to get everyone to work together and to understand each other's language, to write a coherent proposal that brings everyone's perspectives together, essentially.
What were the biggest challenges during the conceptualization phase?
The biggest challenge in a project like this, which is quite interdisciplinary, is always trying to make people understand each other's language. So people that come from different fields often use the same words for different concepts, and it's very difficult in the beginning to try and understand what the other persons are really talking about. So one of the main challenges in a project like this is to bring together all these different perspectives and make sure that we use the right terminology for the right things. And once you get past that stage where we have a concept and everyone understands more or less each other's perspective, then you just have to write it down, essentially, which is still a lot of work, but much easier.
What roles did past research collaborations or gaps in the field play in shaping the direction of PROHITS?
Yeah, so previous collaborations I've already described with Lennart Martens and with Evelyn Peters. So these were the two key collaborations I think that got the project off the ground and the concept. In terms of gaps, there is not that much work done on proteomics on these single cell organisms because it's more difficult. A lot more is done on bigger cells from like human cells or yeast or that are big cells that have a lot of proteins. Prokaryotes and archaea are much smaller and have much fewer proteins, so also then doing these experiments is much more difficult. So this addresses that gap, trying to increase the sensitivity of these experiments and then incorporating that information for essentially the synthetic biology that I talked about, so trying to modify these cells later on to use them in the bioindustry.
What were the main criteria for choosing research institution and industrial partners of the projects?
Yeah, so as I said, in a project like this, you have to have exactly the right people, otherwise it's not going to be funded. So we had to look for people that ideally, as I said, we already knew, that had exactly the right expertise in order to bring them together in the most efficient way so that you have a little bit of overlap, but not too much. You don't want people doing the same thing necessarily too much because you need to have partners that each have their own perspective and expertise that comes together in the right way to bring or to make a complex project like this work.
What was the most challenging aspect of securing funding for the project?
I would say that it's writing the proposal that is the most difficult thing to do, of course. Once you have a really good proposal, you still need to iterate it several times. So it's just a lot of work to try and get it exactly so, to make sure that everyone has given their input and that it's incorporated in the right way in the proposal. But then you need to iterate several times to make sure that there's no loose ends in the proposal, that you try and cover as much as possible. So it's really in the writing. The conceptualization is important. Then it's making sure people can communicate with each other and understand each other's perspectives. And then you have to write it up and iterate the writing. Writing is difficult and always takes a lot of time just to get it to a level that is so good and clearly understandable for people that are not from the project, that they will understand it and that they will understand that it's worth funding. And then you have to be lucky with the review process, because if you get the wrong reviewers, it might still not work. So a bit of luck is always important.
How will you ensure that the project achieves its expected outcome?
I think it's again about the team. If you have the right people in the beginning, then it's almost certain to work to a certain extent. The other thing that was really important and which we have also spent a lot of time on is the interviewing process. So, once the project was accepted, we had to essentially advertise the 10 positions that were available, get all the applications, go through those, order them, rank them, then talk to the people that we thought were the most relevant, and then do more interviewing. So that essentially in the end, and I think this is now that the project has been going for almost a year, I think this is showing that it works, is that so that you get the right people for the project that have the right skills and the right mindset in order to collaborate and work with each other. And once you get those people and the aims are clear, then the project will essentially run itself, which is, yeah. So I think as a coordinator, the key steps are in the beginning. And if you make sure that you spend enough time on that and really get everything together in the right way, then the project more or less runs itself. It's also really important from my perspective, these projects are about the students. So it's also important that the students try and take control as much as possible of their project, and that they connect it to the other people within the project, so that basically everything works already at their level. And then we're of course always there to give input or to help and give advice, but essentially it's them who are doing the work, not us. And it's for them that this project exists as well. Thank you.